evidence why we believe that it's the retinal pigment of LCHAD retinopathy. So advance my slides here. So LCHAD retinopathy, this slide is a montage of retinal images of various The first 4 lines are children who have 1 copy or 2 of the common mutation, 152AC. And this shows the image at the youngest time point progressing to older across the slide. So you can see, you can start to see some pigment clumping and some irregular coloring in this retina at 5 years of age, and quite considerable progression over here at 14 years of age. The same is true for this patient. You can see the dark clumps with hypopigmentation around the areas of pigment clumping. That gets worse, and you see more hypo. Line I'd like to point out to you is this last line here. This is a patient with TFP deficiency, a little bit milder TFP, a little bit later, and does not have the G152AC common mutation. And this retina looks
Melanie, can you hear me? This is Keith.
Yes.
I'm getting some people are telling me they're having difficulty hearing you. Can you speak a little bit louder, maybe, or closer to the microphone?
Sure, I can. Is this better?
Yeah.
Also, everyone needs to mute, really, because if you're typing, then your particular microphone overrides Melanie.
Okay. All right. I'm gonna try to speak up. So the last line is a patient with TFP deficiency, so no copy of the common mutation. And although there is some progression, it's much milder than the lines above. So one of the main points I want to point out is that LCHAD is unique amongst all the fatty acid oxidation. The retinopathy is a unique complication amongst all the fatty acid oxidation disorders. And it seems to be more progressive in patients who have LCHAD or want a copy of the common mutation. So this is the pathway we're all very familiar with. For this presentation, I'm going to define LCHAD deficiency as the presence of 1 or 2 copies of the common mutation G152AC. In fibroblast studies, this mutation is in the LCHAD domain of the alpha subunit, and the hydratase and ketothiolase activities here and here are relatively preserved. They're not 100%, but you do have hydratase and ketothiolase activity with more dramatic loss of LCHAD activity. And I'm defining TFP deficiency as another mutation, either in the alpha or the beta subunit, that leads to mRNA stability or protein misfolding or loss of the entire protein, and therefore decreased expression of all 3 enzymatic activities. So what happens by genotype if we look at their vision over time? This is a figure from a recent retrospective review of about 20 patients. In this cohort, there were 3 patients with TFP and 18 patients with LCHAD. And this is the logarithmic of minimum angle of resolution. So this is when they're looking at that eye chart that you see at the optometrist's office. And you can see that for a logmar of 1.4 to 2, that's considered blindness. Low vision is 0.5 to 1.4 in this range. And then normal vision is up here closer to the top of the graph. In the top graph, we've kind of averaged over across all the patients with LCHAD, the patients with TFP. And you can see that from across the age span, the patients with TFP have relatively preserved vision, while those with LCHAD, defined as one or two copies of the common mutation have a pretty steady decline that was significant over time. If, however, I graph each individual patient, the red lines are the TFP patients and the black lines are the various LCHAD patients, and you can see there's quite a bit of variability. Some LCHAD patients have relatively preserved vision, and some are getting low vision that bounces around a little bit. So one of the things we're interested in trying to determine is what is this variability amongst patients with LCHAD and the decline in vision. However, the graph does pretty clearly illustrate it seems to be more pronounced in patients with LCHAD compared to TFP. So what might contribute to that variability? A while ago, we published a paper that looked at correlations between potential metabolites, toxic metabolites. So hydroxyacylcarnitines was the measure that we used. You could use hydroxy fatty acids as well. Those are a little harder to measure. And we were looking at 13 children with LCHAD and TFP. I think there's only one TFP in this cohort. And we looked at the long chain hydroxyacylcarnitines over a 5-year period of time and how that correlated with their vision at the end of that 5 years. So the Rmax is a measure of retinal function in the electroretinogram. My ophthalmologist describes it as using a foghorn to test hearing. That's kind of what the Rmax is to test vision. You flash a really bright light at the retina and you try to see the maximal retinal response. So a high retinal response was associated with low. This is the sum of the hydroxyacylcarnitines. So this is high acylcarnitines and this would be low acylcarnitines. So the higher the acylcarnitines, the lower the vision. Again, this is correlation, so does not prove cause and effect, but suggests that higher hydroxyacylcarnitines was associated with lower retinal response in the electroretinogram. In the log K is a similar measure that we do in electroretinogram, but again, a good vision or a sensitive retina would be up here, or a high sensitivity would be down here, and low sensitivity would be closer to 1. So the more sensitive the retina, the lower the hydroxyacylcarnitines in this cohort, suggesting that perhaps some of that variability has to do with the accumulation of potentially toxic intermediates, hydroxyacylcarnitines, and or hydroxy fatty acids in the retina that might affect the function of the retina. So one of the questions I had was, are hydroxyacylcarnitines and the genotype that we talked about, correlations, are they related? And I think that's definitely true. So I looked at patients who were either homozygous for G1C, G1528C, or heterozygous for the common mutation compared to those who did not have the common mutation. And I said, what are the hydroxyacylcarnitines in that particular group of patients? So this is after an overnight fast. We fasted patients about 10 hours and we measured the sum of the long chain hydroxyacylcarnitines in this. These are 9 patients, so there's 3 in each group. And you can see that after an overnight fast, the patients that are homozygous or heterozygous for the common mutation have much higher hydroxyacylcarnitines. We fed them breakfast right after the baseline blood sample, and you can see the acylcarnitines come down nicely after a meal, but they don't approach those of the TFP deficient down here. And if you calculate area under the curve of this postprandial response to feeding, you can see that the hydroxyacylcarnitines were significantly higher in the patients with LCHAD deficiency compared to TFP deficiency. So perhaps the retinopathy progression that we see that's more advanced in LCHAD deficiency is a reflection of this increase in hydroxyacylcarnitines. We also looked sort of globally to see where we see retinopathy, and there seems to be less reports of retinopathy in patients from Asia who have less, who don't have the common mutation. So perhaps it's associated with that common mutation, but maybe that's related to the production of these potentially toxic metabolites. we still don't have any direct evidence that it's the hydroxyacylcarnitines that is damaging the retina. We're trying to get that direct evidence. So a couple of questions based on the data so far. We know that if there's several publications that if you can treat these patients, prevent crises, and keep the hydroxyacylcarnitines, hydroxy fatty acids a little bit lower, slow the progression of the retinopathy, but it doesn't halt it. We still are seeing some gradual loss in vision and some changes in the retina. We were curious to know if by newborn screening and we picked them up really early and started treatment, would we stop the retinopathy? At least in our patients that we have followed, it's presenting later. So instead of seeing retinal changes at say 2 to 6 months, we're seeing them in 4 and 5-year-olds. So certainly the early treatment is slowing the progression. But again, we have not halted it. I'm still seeing 4 and 5-year-olds who have started to show pigment changes in the retina. So we clearly aren't stopping the progression in our current therapy. So it kind of led to some thinking about what are the retinal changes? Are they different than other genetic retinal disorders? And in conversations with our ophthalmology team, they say yes, the appearance of the retina in LCHAD is very different than in Leber's congenital amyloidosis or in macular degeneration. It's somewhat punctate. The ophthalmologist describes it as punching holes across the retina in a pattern around the macula. So that led us to ask the question, what retinal cells why are retinal cells in particular affected? What is it about LCHAD deficiency that really targets this particular organ? And what cell type within this complicated organ appears to initiate this progression? And so I'd like to spend the rest of the time talking about the RPE and the evidence that it's the retinal pigment epithelium that might be the cell type that's initially dropping out in this disease. So in this schematic, we have the RPE, this is the retina, and the retinal pigment epithelium is this layer illustrated up here, and it's connected to the rods and cones, the outer segments, these long membranous tails of the rods and cones, which are then connected to the neural retina that send the visual signal to the brain. So the RPE is we believe to be the layer that initially drops out. It's on the other side is the choroid or the blood vessel. So it sits between the blood supply to the retina and the rods and cones or the photoreceptors. If you lose this layer that provides all the nutrients to the rods and cones, ultimately the rods and cones don't have the nutrients or the recycling of visual pigments to function and can die with the loss of the RPE. So ultimately the rods and cones can be lost as well. If we look at it a little bit differently, this is a cross section of a stained section of tissue. You can see that this is the retinal pigment epithelium. It's a nice stained layer here. So they definitely have a pigment to them. They have color to them that gives that smooth red color to the retina. And these are the outer segments of the rods and cones that are attached to the RPE. And then down here is the choroid. So the blood supply is down here feeding the RPE, and the RPE passes the nutrients to the rods and cones. It also recycles these outer segments and the visual pigments. So it takes up a lot of the shed outer segments, which are lipid, recycles that, including the retinol and retinal, and sends it back to the photoreceptors. So it's a really key layer of cells that supports the nutrition status and the metabolism of the rods and the cones. So what's the evidence that it is the RPE that dies first? Some of the first evidence was actually published in '98 from an autopsy by Dr. Tiney's group of a deceased 14-month-old male with LCHAD. So here is again that segments stained from this autopsy. And you can see places, this is the retinal pigment epithelium, and there's arrows to show places where it's missing. So there's a loss of RPE right here. And then there's macrophages, these large round cells, macrophages that have infiltrated this RPE layer. And Dr. Tiny's group suggests that as the RPE dies, macrophages move in to phagocytize the debris within the retina, and that the RPE appeared to be the first layer that disappeared. Here's the choroid down here, the blood supply. She later went on and did some immunohistochemistry staining of cadaver eyes and found that all of the mitochondrial trifunctional protein subunits are expressed in the RPE as well as the rods and cones, but are less expressed or not expressed in the choroids. So it really appears that the RPE has strong staining for both the alpha and beta subunits of TFP. So what are some other evidence that it is in fact the RPE that is dropping out first in this progression of this disease? These are color photos of two different LCHAD patients, A and B on the top, and then a wide field autofluorescent image on the bottom. The autofluorescent image picks up specifically that pigment that is in the retinal pigment epithelium. So the smooth dark areas are the pigment that in the RPE, and the white areas is a loss or absence of RPE. And you can see that you have these spots where there's no pigment observed or blood vessels underneath in this retina, whereas this one looks a little bit more preserved with more RPE, but maybe some spots that are starting to disappear, correlating with those white spots right up here. So the autofluorescence in a number of patients that we've done suggests that there's extensive RPE loss in the posterior pole. And that associated with that loss, there is also a dropout of the choroid or the blood vessels that supply that area of the retina. And this progression seems to start centrally and move towards the periphery of the eye. One of the really cool new technologies is optical coherence tomography, or OCT imaging, where you can actually image the layers of the retina in vivo. So these are two different patients with LCHAD deficiency who we were able to get OCT images on. So in these images, I'll try to walk you through it. The RPE is this layer right here that should be kind of a smooth, dark layer, and you can see some spots where there's no pigment showing up. And the rods and cones are the top dark layer here. The arrows are pointing to some curled retinal tubulations in the rods and the cones, both here and here. And the retinal tubulations correlate to areas where the RPE has been lost. There's no pigment underneath it. So retinal tubulations are when the rods and cones curl up on themselves. And this is seen in a few other diseases that are bottom-up type of disorders, where the rods and the cones are attempting to survive by curling up and trying to provide nutrition amongst themselves, but they're not getting any sort of nutrients, either glucose or recycling of outer segments from the choroid. So in an attempt to survive, they sort of curl into these tubulations. And this is a characteristic feature of a disorder where the choroid and the RPE are lost before the photoreceptors. In the literature, there's about 4 patients that have had this imaging that have advanced LCHAD retinopathy, and all of them have reported this retinal tubulation observation, really suggesting that the disease is starting below the photoreceptors in the RPE or potentially the choroid, but we suspect the RPE, and that's ultimately causing the loss of the photoreceptors above. So this is a complicated slide, but I just want to talk you through it, how we'd like to go about definitively showing that it is the RPE that is the cell type that's dropping out first. Panels A through H are one patient with LCHAD deficiency. K through R are a second patient with LCHAD deficiency. And I'd like to spend most of the time just talking about H and R, these panels here. So the blue outline is the remaining photoreceptors. So if you look along the blue, you can see this is where the photoreceptors are still intact when we mapped them by OCT imaging. When you look at the green is the remaining area of the RPE. So when we used autofluorescence, we overlaid the remaining RPE. And you can see there's RPE that's not underneath these photoreceptors over here. And the red is the remaining choroids. So where the blood vessels illustrated in this network here are still supplying the retina. So it looks like there's areas where the rods and cones do not have support of RPE and choroid, and suggesting again that these cell types are dropping out first. So again, the blue is outlining areas where rods and cones are still intact. The green areas where the RPE is still intact by autofluorescence, and the red where the choroid exists. So suggesting again that it seems to be the RPE and the choroid that are dropping out before the rods and cones. So what are the implications? If we were, hope I've convinced you that it's the RPE that's probably the initial cell later to drop out during the progression of LCHAD retinopathy. What implications does that have for the future of the disease? One of the things I still struggle with is why the loss of the RPE? What about LCHAD deficiency affects this cell type? And is this a cell type that could be targeted for treatment or prevention of LCHAD retinopathy progression? The retinal gene therapy that was just approved in the United States actually treats the RPE for Leber's congenital amyloidosis and replaces the RPE65 protein in that cell type. So there are options that are being used to treat RPE in other congenital retinopathies. So that's the background, and I'm going to stop there and take any questions before I turn it over to Tiffany to talk about how we've moved forward with trying to model RPE in vitro as a way of testing this hypothesis.
Melanie, those changes all occur with or without DHA. Does the DHA slow them down?
Great question. The DHA does not seem to slow down the loss of the RPE or the pigment changes. When we supplemented with DHA, it sped up the speed in which the retina responded in the electroretinogram. Very similar to preterm infants who have DHA deficiency. If you supplement them, then the retina gets faster. But ultimately, over time, once the RPE dropped out and the photoreceptors were lost, then the DHA had no effect. So DHA deficiency will slow it down, but it doesn't prevent the progression. And
this may be a question that should be held till after Tiffany, but are there any enol-CoA hydratase inhibitors that can be used to test whether or not blocking transition to the metabolism to the 3-hydroxy acyl CoAs is helpful?
That's a great question. You might know better if there's a hydratase activity or a hydratase inhibitor. So yeah, that would be an interesting question. The other question we've kind of toyed with is, are the hydroxyacylcarnitines or fatty acids being produced locally in the retina, or is it coming systemically? So if we lower them systemically, does it have any effect in the retina? I guess the data that if we prevent their crisis, slows it down, suggests some of it's coming from the periphery. But I wonder if some of it's actually being made locally. In other words, you have
and if you had an inhibitor, you would see it. You would see it locally, and you might see it in your culture system. So that's why I was sort of saying we might hear a little bit more from Tiffany. Waleed, I see you online. Do you have any? Have you ever run across anything that looks like it might be a hydratase inhibitor? Well, Leeds in Osaka, so we're having a really um, spread out lab meeting here.
Any?
I'm sorry, I was muted. Yeah, I did not find in the literature something about an inhibitor for the hydratase activity, but I suspect it's also going to be a little tricky because of the reaction itself is has to be it may be it can target other proteins. That's what I'm trying to say. So, but I'll look it up.
Okay. Any other questions about RPE? Did I convince you, Jerry, that it was the RPE that's dying first?
I think it's pretty intriguing, Melanie. You know, it's certainly worth following up on, and I have no idea what the heck that NIH study section was grousing about.
So I think there's still out whether it's the choroid or the RPE, that those two layers are so integrally attached that at least amongst our group we've still been debating. Is it the blood supply, the choroid, that's falling out before the RPE?
Right.
But, since the choroid does not express TFP very much, we've kind of looked towards RPE. So, all right, I am going to switch chairs with Tiffany, who's going to talk about how we've moved forward with this idea that it's the RPE that might be the cell type that we want to target in any sort of therapy.
Hello, can you hear me pretty well?
Yes.
Okay, great. My name is Tiffany Devine, and today I'll be telling you about some of the work that we've done towards modeling LCHAD retinopathy in our patient-specific cell line. So to start out, I just wanted to briefly discuss some of the goals of the project and lay out why we're creating the cell model. So as Dr. Gillingham noted, we still don't fully understand the pathology of LCHAD deficiency in the retina, and we're hoping to gain some insight into that with this cell model. So ideally, we'd like to recapitulate what occurs in vivo, and if this turns out to be successful, then the cell model could be used as a tool to test future therapeutics. And as Dr. Gillingham's mentioned, we're particularly interested in maybe a possible gene therapy approach. And so for that, a human cell line would be extremely helpful. So some of this has already been covered by Dr. Gillingham, but I did want to highlight just a couple of things here. So she mentioned that the RPE is really sort of integral in maintaining the health of the outer retina. And so you can see here, oops, sorry about that. You can see here that in this schematic that the rods and cones interact with the RPE, and it's here that they phagocytose the outer segments. And so this is occurring each day with the onset of light, and the photoreceptors are shedding approximately 10% of their outer segments. And importantly, these are enriched in both saturated and unsaturated fatty acids. So the RPE are really dealing with a large load of fat on a daily basis. So we started out with patient fibroblasts that were homozygous for the common mutation G1528C, and these cells are reprogrammed into induced pluripotent stem cells using a four-factor approach. And this cell type has stem-like properties, and it's capable of being differentiated into virtually all primary cell types. And so one nice advantage of these cells is that they can be passaged quite a few times. And so we actually go back to this population of cells in order to derive the terminally differentiated cell lines, because often these terminally differentiated cell lines can only undergo limited passaging. So individual iPSC clones are then exposed to various growth factors and small molecule inhibitors over the course of a 14-day period. And we end up with a sort of a precursor population that's not entirely homogeneous, but almost entirely RPE. And then over subsequent passages, the RPE cells are purified out. And so that we normally assay the cells at passage 3 or 4, but by that time we have a completely homogeneous population of RPE. And one thing I didn't mention was that if you continue to passage these cells for too long, that they start to lose their epithelial characteristics and they'll take on a more stromal phenotype. So it's really important to kind of use them around passage 3 or 4 and always go back to the iPSCs to rederive the retinal pigment epithelial cells. So when we initially plate the cells, they have a stromal phenotype and they grow to confluence and then basically sit there and start to mature and take on a more epithelial phenotype. And this takes roughly 4 weeks to occur. So here I'm showing you some of the immunofluorescence staining that indicates that we do indeed have retinal pigment epithelial cells. And so over here on the left are the wild type, and then here on the right are our LCHAD lines. And these are just sort of various markers. ZO1 stains the tight junctions, and you can see they have a nice epithelial phenotype. BIDF is a transcription factor that's important for this cell lineage. And then RPE 65 was mentioned earlier, which is involved in recycling the retinol. So the next thing we looked at was the expression of different FAO enzymes in our cell lines. And previous work has established that fatty acid oxidation enzymes are expressed in the retina. And this is interesting because it had previously been thought to be mostly glycolytic. So consistent with the staining that Dr. Gillingham showed earlier from Dr. Tiney's group, we also find positive staining for both HADHA and HADHB by immunofluorescence in wild type and LCHAD deficient cells. And then over here on the right, I'm showing you some Western plots for you can see again, the MTP subunits are nicely expressed. I'd also like to point out that HADHA appears to be relatively stable in our LCHAD deficient cell line. And RPE are known to express the 1A isoform of CPT1, so that's this lower doublet here, I believe. And then on the right, I'm showing Westerns for which acyl-CoA dehydrogenases are expressed in RPE. And we find a robust expression of ACAD9. So here I've included 293 cells as a positive control for the antibodies. And VLCAD may be expressed at lower levels, but I would probably caution from definitively making that conclusion, because it's possible that this antibody isn't great for Western, because I've seen it expressed much higher in 293 cells. But in any case, I think we can say that either ACAD9 is the prominent ACAD, or that it at least overlaps with VLCAD. And this is interesting to us because it provides some explanation as to why VLCAD patients may not develop a retinopathy. And perhaps it's because ACAD9 can compensate for the loss of VLCAD and RPE. Okay, so one diagnostic hallmark of LCHAD deficiency, as you know, is an increase in long-chain hydroxyacyl-carnitines. So in this experiment, mature RPE were fed either BSA or BSA palmitate for 48 hours, and we then collected media for acylcarnitine analysis. And you can see here that so C16 is the dark bar, and then the hydroxy C16s are the lighter gray bar. And over here on the left is the wild type. So this is in the presence of BSA and BSA palmitate. And you can see there's a pretty dramatic increase in the hydroxy species in LCHAD deficient RPE. So this is kind of consistent with some of the data that Dr. Gillingham showed earlier. All right, so we looked at oxygen consumption rate as well in both wild type and LCHAD deficient RPE using the seahorse. So in this experiment, the RPE are exposed to either BSA or BSA palmitate at the start of the experiment, and we normalize the OCR values to the baseline measurement, which is set to 100%, and the black dotted line here shows that wild type RPE increased their oxygen consumption, whereas we did not observe an increase in LCHAD deficient cells. So this indicates that they're not able to utilize long chain fatty acids as an energy source. Okay, so earlier I mentioned, and Dr. Gillingham also discussed, that one of the primary functions of RPE is to phagocytose these outer segments, which are enriched in fatty acids. And so we can also do this in culture. So here we can isolate bovine outer segments. We can label them with FITC, and then they can be fed to RPE. So in this experiment, they're fed to RPE for 3 hours, and then we fix the cells and we can analyze them. And so what we find is that the RPE over that course of time will take up some FITC labeled outer segments, and some will remain on the surface. If we then label the cells with rhodopsin, so these outer segments should stain positive for rhodopsin, without permeabilizing the cells, we'll see double staining on the surface. And if we see green staining only, that indicates that the cells have ingested the outer segments. And so you can see here both wild type and LCHAD, our cultured cells are able to phagocytose outer segments. And the yellow, the double staining is surface bound outer segments. And then over here, this is just a confocal image. This is from LCHAD RPE. And so here you can just kind of get a look at the a different section of the cells. And so these white lines correspond to the images found over here on the sides. And you can see that the green dots out here on the edges are outer segments that are still stuck to the surface, but you can clearly see that some of them have been phagocytosed into the cell. And so these we can actually visualize in the cell. So overall, this suggests that our RPE are functional, and that they can take up outer segments. All right, so this is sort of switching gears a little bit here, but I just wanted to discuss some relatively recent data that suggests that RPE can, in addition to being able to use fatty acids for energy, these acetyl-CoA generated in beta oxidation is also a substrate for ketogenesis in retinal pigment epithelium. And it's shown that some portion of the ketone bodies are exported from the apical domain, and they support the outer retina. So in this particular experiment, we assayed media collected from the apical chamber of a transwell system for the presence of beta-hydroxybutyrate following exposure to either BSA or BSA conjugated to palmitate. And we found that the amount of beta-hydroxybutyrate released was reduced in LCHAD deficient cells. So this suggests that if they are releasing fewer ketones, then this may affect the health of the photoreceptors. Another feature of RPE being a polarized cell is that they secrete particular factors preferentially either apically or basally. And so two examples of this are that they secrete VEGF basally to support the choroid, and PEDF, which actually inhibits VEGF, that is secreted into the outer retina. And so here we've done an ELISA to look at VEGF secretion. And this is a common assay that's done to test the functionality of RPE. And actually what we found was that in general, LCHAD deficient cells seem to secrete less VEGF. And so they do secrete some basally, or some apically, I'm sorry, but most of it's basal. And it looks like in both cases, it's decreased in LCHAD deficient RPE. So I just wanted to share one last experiment. So Dr. Gillingham mentioned earlier that one intriguing hypothesis about the pathogenicity of G1528C in RPE is that the metabolic intermediates generated during beta oxidation may have a toxic effect on surrounding cells. So in light of this, one question we're interested in is whether wild type cells can clear the long chain hydroxyacyl carnitines produced from LCHAD deficient cells. And if so, what proportion of cells would need to express the wild type enzyme to effectively clear these species from the local environment? So here we fed both, so the blue circles indicate wild type and the green circles are LCHAD deficient cells, sorry about that. So here we fed both cell types, either BSA or BSA conjugated to palmitate. We then collect the media 24 hours later in both cases. And then we took media from LCHAD deficient cells that had been exposed to palmitate for 24 hours and placed that onto wells with wild type cells. So we completely replaced this media. And then we sent all of this off for acylcarnitine analysis. And so the red boxes are data that you've seen before, or consistent with data that you've seen before. So in this case, wild type do not produce much do not produce C16 hydroxys, whereas the LCHAD deficient cells produce quite a bit of C16 hydroxy when exposed to palmitate. But interestingly, when we put that media on top of wild type cells, we're able to clear the hydroxy, the C16 hydroxys. And so in future experiments, we're going to be doing probably some co-culture with both wild type and LCHAD to sort of determine what percentage of correction or what percent of wild type enzyme would need to be present in order to clear these hydroxy species. So just to sort of summarize what we found so far. Again, the RPE is an extremely important cell type for the outer retina. It acts as a barrier and passes nutrients through. There's data to suggest that RPE, after phagocytosing outer segments, can use the fatty acids from these outer segments either to produce energy for itself or to produce ketones that are then secreted into the outer retina. And in our LCHAD deficient cells, we find a decrease in oxygen consumption when exposed to palmitate, as well as a decrease in the release of beta hydroxybutyrate. And finally, we're also finding a pretty substantial accumulation of long chain 3-hydroxyacyl carnitines in our cell, and a decrease in the secretion of VEGF, which could affect the underlying choroid. And so with that, I just wanted to thank Dr. Gillingham for letting me do this work in her lab, and also thank Garen for creating the iPSC lines and working to create more. And then, of course, the Harding lab for all of their feedback. And then Dr. Return's lab has done the acylcarnitine analysis for us. And with that, you can take questions.
Tiffany, that was great. I have some questions, but before I do that, and while I still have everybody online, I'm going to remind you all to register for the upcoming Inform meeting in Athens, and especially if you can send. Whoops, I'm getting a lot of feedback. If you can send folks from your lab, we would love to have all of you attend, and there are opportunities for trainees to get travel awards to attend. So please plan on being in Athens. Now, I did have some questions about your cell system. First of all, do you see any cellular signal of increased turnover, apoptosis, cellular stress when supplemented with long chain fat substrates versus just fed on glucose, for example?
So right now, can you hear me? Yeah. So right now, what we've tried is a somewhat long-term feeding with palmitate. So I went up to 5 weeks with palmitate, and I did not see a lot of cell turnover. I can say that these cells are extremely robust. They're resistant to pretty high levels of even hydrogen peroxide. But that said, we've kind of changed approaches at this point, and we're now starting to feed them the outer segments, which we think will be a little bit more physiological. And we've chosen an endpoint of around 3 months. And so we're going to go ahead and decide kind of what assays we want to look at that time point if there's not a significant amount of turnover. So yeah, it's been a little bit tricky thus far in terms of trying to kill the cells with exposure to fatty acids. And it may just be something that's going to take quite a bit of time. So there are other examples in the literature where groups have used these types of cell lines derived from patient cells. So
given the length of time that you might need to see a phenotype, can you do some parallel experiments with some other cell lines? For example, if the difference is in the 3-hydroxy versus the overall, you should be able to take TFP cell lines, cells differentiated to RPE, and look for a physiologic difference or a biochemical difference in the two of them. And barring that, you could knock out the beta gene in your wild type iPS cells and create a model. It might be nice to do those side by side, especially if you're talking about 3 or 6 month experiments.
Yeah, absolutely. So another option is we were also thinking about looking at VLCAD. So we're in the process of creating those iPSC lines right now. We also have some TFP lines some fibroblast lines that we've talked about probably pulling those out and creating iPSCs as well. So yeah, both excellent suggestions.
I'm monopolizing. Other questions for Tiffany or Melanie?
One question, you showed that you'd done the oxygen consumption with C16. Did you try medium chain fatty acids to see if they're utilizing the medium chain better?
Yeah, that's definitely on the list of experiments to do. So we have talked about that. Yeah, it would be an excellent positive control to show. It's here in the future. Yes. The seahorse experiments aren't always as straightforward and easy as I would like them to be, but yeah, the medium chain experiments are something we've discussed and plan on doing.
Are you planning to do medium chain like a triglyceride or include it, or just straight the fatty acid straight?
I think just the straight fatty acids. Yeah.
Octanoate,
Yeah.
BSA.
We have octanoate. Yeah.
Yeah. Yeah, in cells you would just do the isolated fat, because that's when, you know, in the bloodstream that's what you get.
Could you try something else, like different energy providing molecules? Have you thought about it?
That's interesting. Yeah, so there's a couple of interesting papers that just recently came out that show the RPE actually will sort of forego glucose and pass it up to the outer retina, and the outer segment, or the rods and cones, will then use the glucose and then provide lactate as a substrate to the RPE. So there are a couple of options there, and we're sort of interested in looking into some of that glucose pass through and utilization of lactate. Yeah.
Given your VLCAD findings, might you want to use an ACD9 deficient cell line for your studies?
That would be wonderful. I mean, right now we only have a VLCAD line, I think. Yeah, but yeah, that would be.
Yeah, well, we have ACD9, and Melanie, we ought to talk, because we want to make some of these as well. So IPS cells. So you know, we might talk about splitting which ones we do and sharing, so that we can also share the cost. The other question is, you know, Arig or Gabali from our group has shown this difference in cardiolipin content in the MTP versus LCHAD, with the cardiolipin defect being more pronounced in the LCHAD deficient cell lines. It would be interesting to look at your RPE cells and see if that translates. Those are all on fibroblasts, so it would be interesting to see if that translates to the RPE cells. So if you have cell pellets, I don't know how robust these are in growing, but if you've got cell pellets and you can send them to us, we could test those.
I can't hear Grant.
Grant, you didn't come across. Either type your question I think your microphone was unmuted, because I could hear sound, but you sounded garbled. You're muted now, Grant. I can see that. We can't hear you, Grant. Sorry. You still look like you're muted on according to my screen here, so I don't know what's going on. All right. Well, I think we're out of time anyway here, folks. I actually have to go back to my meeting. I'm supposed to be teaching right now at the SSIM Academy. So one of my partners is taking the first 15 minutes of this to give me a free. Anybody who would like to do next month, we have an opening. I will send around an email inviting the participation. And otherwise, I'd like to thank Melanie and Tiffany for taking this on. I think it's a great project. And Melanie and Tiffany, you and I need to talk a little bit more about some of the biochemistry and some of the cell studies. All right, bye-bye.
Thank you very much. Thank you, Melanie. Thank you, Tiffany.
Thank you.
Yes, thank you.